We love our museums here on Tested, and one of the most important and august museums in the country is right here in San Francisco, our own California Academy of Sciences. It was founded over 150 years ago and is not only a world-class museum, it is still an active and important research facility. I recently visited their Institute for Biodiversity Science and Sustainability, the Academy's research arm that is home to over 46 million specimens from all over the globe. I visited the back stacks of the Academy to learn the history and the care of these amazing collections and how researchers are scanning the collection digitally with modern scanning technologies, all to help the public better understand evolution and the interconnectedness of organisms and ecosystems. Lauren, tell me exactly where we are right now.
So we are in the Herpetology, so the reptile and amphibian collections at the California Academy of Sciences.
Okay. Now, there are parts of this collection on display for the public to view. Why is there so much not on display? What are all these doing back here?
So these are mainly for researchers. This is a historical collection. We have things from the mid-1800s until now, and we have specimens from like about 174 countries.
How many specimens in total are there, maybe in herpetology and then in general?
So in my collections, there are 320,000 specimens. As a whole, in the museum, I think we're going with about 46 million.
Wow. Okay, so with all of those specimens, is most of your time just spent documenting what you guys have, or?
A lot of it.
Tell me about how this collection moves around, what its main purpose is.
So I describe my job as like a librarian of dead things. So I do a lot of loans. So I receive and I send out snakes in the mail. For researchers, I have visitors who come in to look at them. They take them out of the jars, they measure them. I also maintain a tissue collection. So I send tissues away for molecular work. And then we also I'm doing a lot of cataloging. So we maintain a database. So every specimen has its own unique number, and everything, all the information about it is online.
And you guys aren't just storing like one of everything. You have many of some things, is that correct?
Yeah, each specimen is kind of like a snapshot in time.
Yeah.
So it came from one time, one place, and it has its own unique DNA, it has its own unique metadata, and we want to know kind of everything about it. We also want to have many so that we're able to compare kind of across time and space.
Yeah. And this museum has been here for so long. This is an important collection in terms of how far back it goes.
This is a really important collection. The museum started in 1853, and collections were kind of the basis of why we had museums at that point. And unfortunately, our collections burned down in 1906, save for a few specimens. We ended up adopting the Stanford collections. So we still have some of these older collections from the mid 1800s, but I mean, this is what the Academy is really based on.
Yeah. So I want to talk about some of the older specimens, because as we were walking through the aisles, I saw some red tape around the bottles, and that's a particularly neat kind of specimen.
So one of the things that people still do in the collections, here we'll go this way, is describe new species. So it's still happening. New species are being described every year, especially now with advent of molecular work. So
you can be more precise about
You can be more precise, and also there are like cryptic species. So things that may look exactly the same, but genetically they're very different.
Oh, fascinating.
But when someone describes a new species, they designate a single specimen to be what we call the holotype of that species. So that's like the ambassador of the species.
Right.
So anytime you study that, you have to refer back to that single specimen.
Right.
The problem with that is there's no variation. So you have like one male, usually, for a holotype. But you want to be able to show differences in sex, so differences in color morphs, geographic localities, just all the variation. And so at the same time you describe that one, you describe a whole range in that same publication called the paratypes. So those are the red ribbons.
Okay.
The holotypes are blue ribbon specimens, and we keep them separately.
Fair. So when Darwin was in the Galapagos collecting specimens, everything he was grabbing must have been a type.
Pretty much. We don't have any of Darwin's collection, but we do have the world's largest collection of Galapagos species and specimens. And so a lot of what we have been used for research and are type specimens.
Yeah.
Because he didn't go everywhere, and when the Academy went on their Galapagos expeditions, it was one of the most complete or like thorough surveys of the archipelago. So they visited like 24 islands and found a lot of new species.
Well, and this is really particularly like one of the origin stories of the museum. While the collection burned down, you guys had researchers at the Galapagos coming home with specimens that then became the core of the museum.
Yeah, so they knew that tons of species in the Galapagos were in danger. Mariners were coming through.
They already knew.
They already knew it because all of these mariners and these whalers would keep pretty detailed logs.
Right.
And so based on the logs, I think they knew that over 100,000 tortoises had been killed in like a 100-year period.
And the mariners are just keeping these records because it's commerce.
Yeah, and they would bring them onto the boat because they knew that tortoises didn't have to eat and drink much. And so they could just keep them on there and then eat them when they wanted.
Oh my God.
Or they would render their...
That's really bleak.
It is really bleak. Or they would render their fat down and use it for gas lamps. So they used tortoise gas and tortoise oil to light the streets on the mainland.
Right. So even in 1905, 06, when they were there in the Galapagos, they understood what they were doing was this important conservation of information that might be lost soon.
Yeah, they knew some species were already extinct. So they wanted to go and kind of get as many representatives from different islands, different species as they could so that we'd have this historical record.
So do all big museums have collections like this that they are lending to researchers around the world? Is this like, this seems like an entire economy that the public doesn't necessarily always know about.
They do. Most collections have specialties.
Right.
So we have really strong collections in the Galapagos, in Western North America, and in Southeast Asia, and then also Central African amphibians.
You were, before the camera was rolling, you were talking about a local Bay Area snake, and you were kind of, I loved the way you were talking about it. Can you introduce me to this?
I was. So snakes aren't everyone's cup of tea. I have to say I was like a late snake convert, but a really fun snake called the rubber boa.
I didn't know there was a local boa.
There is, and it's what I like to think of as a beginner snake.
It's
So they're harmless. They have blunt heads, blunt tails. They get really wrinkly. And I think it's just a lovely, kind of bland snake.
Yeah, is this about the size of an adult?
You can get some bigger ones, but generally they're not that big.
Yeah. How much backlog is there in a collection like this? How many things are you just trying to chronicle as opposed to maintain?
Some collections have huge backlogs.
Yeah.
I kind of pride myself in this collection in that we don't have a really large one, but it's hard to get data from people. And so a lot of what we have backlogged is stuff that we're waiting on data for or have incomplete data for, because we won't accept anything into the collection unless we have the right permits, unless we have all of the metadata associated with it. So identifications, collectors, localities, because we need to know where it came from.
So I know we used to gather these specimens and store them in all sorts of different things that were not necessarily good for their preservation. So conservation is an ongoing project. I'm curious, when you send these out, is there just a standard international protocol that scientists and researchers follow for handling material like this?
So everything in this collection pretty much is in 70% ethanol. So like drinking alcohol.
Yeah.
A lot of it was preserved with formalin, but then that is absorbed and then washed out and then it's stored in ethanol.
Yeah.
And so we just have to follow kind of the rules, the mail rules to send small amounts of alcohol, which I think changed a little bit with hand sanitizer.
Oh, wow. Look, did it get a little easier?
I think it got a little easier, but there are a lot of regulations
Yeah.
in terms of sending things internationally. And so we have to go through a lot of permitting for that.
But I mean, I was also assume that, like, are there sometimes you send a specimen out and a researcher does something you didn't expect, like cut it in half, and you didn't know that was going to happen?
Ideally, that won't happen. But people have to send in requests and like pretty detailed summaries of what they will do with the specimens.
Right.
And we have to give them explicit permission for destructive sampling.
Yeah.
So we just hope we get them back.
So talk to me about the digital aspect of chronicling and maintaining this collection.
So some of the things that we have, we can't send on loan. And sometimes we can't send loans to certain countries, to certain individuals. And so we're trying to digitize a lot of the collections to make them more accessible through photographs and now 3D imagery. So we're working on a project now to 3D surface scan and CT scan a lot of our Galapagos collections.
Yeah.
Hopefully, one male and one female of every tetrapod, so non-fish vertebrate, from every island on which they occur. So there are some species that are just on one island,
Right.
and then there are some that are widespread.
How many, is that like 100,000 species or 10,000 species? Or
We're hoping to scan about 1,300.
okay,
Yeah.
that seems reasonable.
Yeah, and we're at like 900 now. And that's, there are some that have multiple, but we're working on it. So
I'm curious, like, when you get to, is there a moment when you like break out the champagne, like we got the whole collection digitized? Is there a party?
I think it's when the NSF money runs out.
Fair enough.
Yeah.
Do you have favorite specimens that like you go back to or that remind you of why you started doing this?
So I do have some favorites. I also have some favorite types, like not species types, but
Sure.
kinds of specimens.
Yeah.
And I always love to show off our cleared and stained collection. So this is something that we don't do as much anymore,
Right.
especially with CT scanning, because this is a pretty destructive way of
Being able to see the
able to see the insides of a species or a specimen, but
But it's really cool.
it's
looking.
really cool. So for this specimen, you take the skin off, you eviscerate it, but all of the muscles are still there.
Right.
And so you can kind of see them, but they're cleared. So when you
Yeah.
take it out of the jar, it has the consistency of like a soggy gummy bear. And it's kept in glycerin, so they're like a terrible mess.
Right.
But they're dyed twice, one with red dye to look at the bones, and one with blue dye to look at the cartilage.
And so they're pushing these dyes through it, and only those two. There's a certain like the tissue is absorbing only one color.
Yeah.
Okay.
And so if you were to just skeletonize this, a lot of it would just fall apart. And you wouldn't get the things like the trachea or like the bits in the eyes. And so it's a really, I think, beautiful way of preserving a specimen.
But nowadays, that's what the CT scanner can do that we don't have to do this level of destructive.
Yeah, and so you can, with a CT scanner, you can look at the hard skeleton, or you can do kind of contrast iodine staining and look at soft tissues as well.
Amazing.
And without like taking the skin off of the specimen.
Yeah.
There are some things that you can only really do with these. So it's really good for tadpoles. So before, as they're developing, they haven't keratinized, like their bones haven't hardened.
Right.
And so you can see a lot of structures with this type of staining that you wouldn't be able to in a CT scanner.
That is really cool.
Yeah, I think they're great.
They look amazing.
Jack, tell me what we're looking at here.
This is a medium ground finch, so it's one of the Galapagos finches, or the aka Darwin's finches, right? And they're really well known for the evolution of their bill size. So there's the small ground finch, the medium ground finch that we're looking at, and large ground finch, and they sort of specialize on different food types.
And this is beak differentiation even within islands on the Galapagos.
Right, exactly. So sometimes you'll get 2 different species on an island, and occasionally in a couple of the largest islands, you may even get 3. And it's largely because they're divvying up, the food. They're eating different things, and that allows them to coexist. Because if you're eating different things than me, then we can both live on the same island. But
we're not competing.
Exactly. And so, and interestingly, like the evolution of the finches and the finch bills have played really heavily in evolutionary biology. Some of the early papers on community ecology by David Lack, were testing ideas about, how different do they have to be to coexist, and can 2 different species be each other's biggest selective pressure. And then the Rosemary and Peter Grant are famous for having actually shown the evolution, because they had data from multiple years, and an El Nino hit and you could actually see the bills changing size.
Wow.
Which is amazing, because you usually look at like pictures of horses' hooves, and it's like, 60 million years ago they look like this, and 30 million years ago, so you think, you'd never notice evolution, right? Because it's like so slow, it takes millions of years. But it's actually happening, it actually does happen really, really rapidly, and you can see those changes if you take the right measurements.
Now, just the samples that Darwin and other expeditions brought back from the Galapagos help tell the story of evolutionary biology. I'm curious if the scan helps deepen that story.
Well, so, one of the big questions is like, how did the beaks evolve? What factors have changed? And so in recent years, people have sequenced the entire genome of the Galapagos finches to figure out, well, which genes are getting turned on, which ones are getting turned off? How is that actually manifesting? So trying to understand that evolution and, and how turning on these genes and turning off these genes actually changes the morphology is a really important question, how these things all work. And so when you have a CT scan like this, you can actually see all the different bone elements in really high detail, and you can actually see what's changing. And so if you had the genome, you could actually, try and see what's going on.
I mean, digitally, you could actually take two different skulls and put them on top of each other to see their differences too, yes?
Yes. In fact, John has done that with a couple of these medium ground finches.
That's right. That's what we prepared here. So we took a couple of different scans of different specimens of the same species. Here, in this kind of view, the color denotes relative density. So the bluer areas are less dense. That would be the flesh and musculature and so on. And then the red areas are more dense. So that would be the beak and the skeletal structure.
And these guys are missing most of the torso skeleton. Is that correct?
No, these guys got it.
Oh, these guys have it.
Which is exactly why we wanted to see these specimens.
These are wet samples. And so these have been preserved in ethanol. And you can even see, Jack would know better, but I believe this is even the stomach contents.
Yeah, that's probably his last meal.
Yeah.
So whereas the wet samples are poor for preserving color data of the original thing, they're spectacular for exactly this.
Exactly. So you can, if you can CT scan them, then you can get all the data from the skeleton. And in fact, there's ways to stain the muscles so that they become, more opaque to
within
the X-rays.
The scan.
Yeah. So you can stain them with iodine or osmium tetroxide, and then when you scan them, you can actually see all the muscles because the others, the specimens that we have, the skins, they take everything out of the inside, and so all you have left is the skin, the feathers, But that's important because you can see the colors and how we tell the different birds apart. But if you want to find out what's going on inside,
well, and regarding that, I mean, normally microscopy would allow us to take these things and enlarge them so that we could investigate them. But is there a utility to taking a scan like this and printing it up in a large scale to understand better the bone's topography?
Yeah. Oh, yeah, absolutely. In fact, you don't even have to print it. You can actually just blow it up on the screen and spin it around and you can see it. But, one of the things that we wanted to do with some of the scans that they've been scanning for this project, the OVERT project, is actually to have the Darwin's finches bills, because then you can print them out and you can print them at whatever side you want, and kids can use them in schools for education. Because most people don't get an opportunity to see Darwin's finches.
Right.
Mm-hmm. And if you had little plastic 3D printouts, you could actually see them and hold them in your hand and see how the different finches vary.
Well, it just occurred to me that the differentiation of the beak would be about the food type that it's eating. You could almost do practical demonstration with kids about what it's able to go get or obtain.
Yeah. And, in life, they've done these cool experiments where they created a little tool that measures bite force. So as it bites down on this little stick, they can actually measure the number of pounds or, kilograms it exerts. And so, you could probably do the same thing with the plastic printout and see how far it goes before it breaks.
Right.
And of course, it wouldn't be exactly the same because it's plastic instead of bone, but it would give you some idea.
As a scientist, is there an aspect of this technology that allowed you to see something that you hadn't formerly been able to see?
Yeah, there was one that you had scanned that we could see some weird things on the skin, and I wasn't, honestly, I don't know what they are yet. One of the interesting things is that, these birds do get avian pox, and usually researchers find it by looking at the exposed skin patches, the feet and the face. And I don't know if these are avian pox lesions, but one of the things that's cool about the CT scan is it's looking through the feathers. So if there's lesions on other parts of the body, you might see it. And in other work that they've done here and with colleagues where they've scanned a lot of herpetology specimens, they find internal parasites. You can find little worms, you can find other pathologies, things that you can't see from the outside of the bird. And, and then, you know exactly where it is. So if you wanted to, you could make the incision, go in and get that worm and figure out what it is. But otherwise, you'd never find the thing.
That's it. You said non-destructively.
Non-destructively, you can figure out what's going on. So that's really cool.
Yeah. And you go to the parasitologist lab down the hall? Or no, it's not one. Is there?
No, actually, we do. We have a veterinarian who works at the aquarium here.
Yeah.
And so we often find weird things in birds and mammals when we're skidding them. And I often go down to talk to
That's
Freeling
neat.
and say, Freeling, what's going on? What's this weird looking thing?
When you're doing this research, you're building grants and research avenues to try and look at certain things. But when you come across something like this, you don't know what this is. How would that eventually, would you eventually like put that off to the side? Like maybe I'll write about that later, or you'll research and figure out what it is and write a small paper about this artifact being made clear with this process?
Well, you know, that so many times you'll see weird things, but if you see weird things over and over, you start to scratch your head and say, what's going on? What's special about those specimens? Like let's go have a look at those things. And it's often when you start seeing patterns that because, I mean, we see so many weird things every day. You work in a collection like everything is kind of weird.
Yeah.
Right? Yeah. So trying to figure out what are the interesting things. For example, one of the things I'm known for is studying poisonous birds. And,
Poisonous birds?
Yeah, poisonous birds. And we didn't realize, the first time that I got poison on my face from these birds, I thought it could be anything I touched during the day. I could have touched a poison plant, but it's the pattern. It happens over and over, and you're like, okay, something's weird about this bird. Every time we touch it, something weird.
So it excretes a poison onto its skin?
Yeah, it has a potent neurotoxin in its skin and feathers, a lot like poison dart frogs would.
Wow.
But that's the thing. It's like once you start seeing a pattern,
Right.
Then you say, okay, here's something that's worth following up.
And this potentially illuminates whole new avenues for finding patterns.
Yeah, exactly.
Okay.
Exactly.
The other thing that we bookmarked here is something that you were talking about a little bit off camera, and you're welcome to.
Any guesses, Adam, about what that little coil is there? Did he swallow a spring?
What the hell?
Helps him bounce.
No idea.
So that's actually the trachea of the wind.
It is.
Yeah, and those are when you inhale, all that pressure would just cause a fleshy tube to collapse. So what birds and we have are these little cartilaginous rings that can become bony in their later life. So
they provide the same structural. They provide the same structure that the ribbing on a vacuum hose does.
Exactly.
Wow.
So that allows them. So that's what they're breathing through. And if you follow that down, you can find their syrinx, which is their noise-producing organ. A lot like our larynx, but they have it a little further down, right at the top where the two bronchi split into the two lungs.
So you can use scans like this to compare and contrast the syrinx of many different.
Yeah.
Right.
Yeah. And birds have these amazing songs, so that would be one way. In fact, the curator of ornithology before me was Luis Baptista, and he spent his life studying how birds make song and how song develops in birds. And he actually has a collection of syrinxes in the back to, because he could study those from birds and understand how they're making the sound.
That makes me want to 3D print it like Dr. Grant from Jurassic Park and make it make sounds.
Yeah, you might be able to do that. That'd be pretty amazing.
That is.
And then these weird blue blobs here, we think those are probably some seeds that it may have eaten. A lot of birds have a crop, or what they can sort of store things. And then when they swallow it, birds don't have teeth for chewing, right?
Right.
So they usually eat the seeds whole, and then they have a very muscular gizzard or muscular stomach that grinds them all up. In fact, some birds will even eat stones to do
like
that
cows,
grinding for.
Cows as well.
I don't know if cows eat stones, but they definitely have an interesting set of stomachs that do crazy things.
So this might not be his last meal. It might be the last meal for his children. Or
maybe, yeah.
But so this is being stored kind of like above the digestive tract. It's waiting to go into the digestive tract.
Right, and maybe waiting to get down into the stomach.
Fascinating.
And another thing that we thought of, there's a fly, a parasitic fly that was introduced to the Galapagos. And we don't know when it was introduced to the Galapagos. And the first specimens we have are from the insect collection in the 1960s. But we have a lot of nests from earlier expeditions, bird nests. So we thought about doing CT scans of those nests
to go look for fly larvae.
To look for fly larvae or the fly pupae.
Are you guys starting to think about using AI to be able to parse large volumes of certain features through a collection?
Absolutely. This is the cutting edge of this type of analysis now, is that you can use AI to interpret the scans. And so we actually have an assistant in our software. It's called Atlas, that you can speak to. You can ask it questions about the scan that you've run and say, hey, what's going on with this scan? What am I looking at? And it'll ask you a few follow-up questions and then sort of interpret the scan for you and make it easy to figure out what's gone wrong in your manufactured product, or what's interesting about something else that was scanned.
I love the fact that the original collectors of these specimens had no idea how deeply we would be able today to look into them, but they had enough rigor that these are still useful to us because of the data that came along with them.
What I'd love to think about is like what people will be doing with our specimens 200 years from now. And it's a really good case for collecting as much as we can to document what's on Earth right now, because it doesn't just document, that individual or, there's this species that occurred in this place, but it's like its last meal. It's, you can get stable isotope signatures and figure out what its diet is, based on that. You can DNA and RNA. And, a lot of these specimens were collected long before anyone even knew what DNA or RNA was. And so it kind of blows your mind and you think, what are people going to do 200 years or 500 years from now? And and that's why it's so important that we preserve all these specimens.
What an incredible resource. Yeah.
That's really
it's like a library of life. Yeah.